Today we are going to learn some aspects of immunology like immune response during mycobacterium tuberculosis, malaria infection, HIV, and immunodeficiency, along with a small introduction about vaccines. Delayed type hypersensitivity response in case of mycobacterium tuberculosis. The delayed type hypersensitivity response does not correlate with protection against TB. Since numerous BCG vaccination trials have demonstrated that disease can occur in those who mount a DTH response. As a result, the protective T cell response must be distinguished from the T cell response associated with DTH. An in vitro interferon gamma release assay has been developed. The assay is an alternative to the tuberculin skin test (TST) for detection of latent M. tuberculosis infection in human hosts. Then immune response during malaria infection. Additional components of naturally acquired immunity include the release of cytokines that act against all stages of the parasite and also a cytotoxic T cell response directed at liver stages of the parasite. Then antibodies block invasion of sporozoites into liver cells. IFN Y and CD8 T cells inhibit parasite development in hepatocytes. Antibodies block invasion of merozoites into erythrocytes. So antibodies block invasion of sporozoites into liver cells. IFN and CD8 T cells inhibit parasite development in hepatocytes. Antibodies block invasion of merozoites into erythrocytes. Antibodies prevent sequestration of infected erythrocyte by preventing binding to adhesion molecules on the vascular endothelium. IFN gamma and CD4 plus T cells activate macrophages to phagocytose intra-erythrocytic parasites and free merozoites. Antibodies neutralize parasite glucosyl phosphatidylinositol and inhibit induction of the inflammatory cytokine cascade. So antibodies mediate complement-mediated cytolysis of extracellular gametes and prevent fertilization of gametes and the development of zygote. Immune response during HIV infection. HIV transmitted through sexual activity enters the bloodstream via mucous membrane lining the vagina, rectum, and mouth. So macrophages and dendritic cells on the surface of mucous membrane bind virus and shuttle it into the lymph nodes, which contain high concentrations of helper T cells, CD4 plus T cells. Once HIV has entered the body, the immune system initiates anti-HIV antibody and cytotoxic T cell production. However, it can take 1 to 6 months for an individual exposed to HIV to produce measurable quantities of antibody. The immune response is weakened as memory T cells are destroyed. CD4 plus CCRR5 plus. HIV enters the body and binds to dendritic cells, orange cells with the projections, which is on the illustration, which carry the virus to CD4 plus T cells in the lymphoid tissues, establishing the infection. Virus replication accelerates, producing massive viremia and wide dissemination of virus throughout the body's lymphoid tissue. An immune response against virus causes some protection, but a chronic persistent infection is established. The production of cytokines and cell division that regulate the immune response for protection also causes HIV replication. There is a rapid turnover of CD4 plus T cells that ultimately leads to their destruction and to a change in lymphoid tissues that prevent immune response. Cytotoxic lymphocyte production follows the rise of HIV in the blood. HIV-specific CD4 plus T cells may be especially susceptible to attack and destruction by HIV. HIV binds to CD-zyme, a glycoprotein expressed on dendritic cells. Migration of HIV-bearing activated dendritic cells to helper T cells areas of lymph nodes may specifically infect helper T cells specific for HIV peptides. Reductions in HIV-specific helper T cells numbers may lead to decreased activation and survival of cytotoxic CD8 T cells. Reduced CD4 T cells may also result in the incomplete activation of CD8 T cells that can remove HIV-infected cells, resulting in a decreased ability to destroy virally infected cells. The rapid loss of memory helper T cells and the inability to replace these cells leads to increased immunodeficiency. High mutation rates of HIV also allow virus to escape adaptive immune response. Immunodeficiency. Immunodeficiency is a state in which the ability of immune system is compromised or entirely absent to fight against infectious diseases and cancer. Two types of immunodeficiency disorders, primary or congenital, are inherited, secondary or acquired. Immunodeficiency can be specific or non-specific. Specific abnormalities of B and T cells, non-specific abnormalities of non-specific components. Then congenital immunodeficiency is caused by defects in lymphocyte maturation, B cell maturation, and general immunodeficiencies. All are relatively rare, usually present at birth, and are usually hereditary. When evident during infancy or childhood, however, some disorders such as common variable immunodeficiency are not recognized until adulthood. Example, severe combined immunodeficiency or SCID, then DiGeorge syndrome, Chidiac-Higashi syndrome. Severe combined immunodeficiency syndrome, congenital immunodeficiencies that affect both humoral and cell-mediated immunity are called combined immunodeficiencies, characterized by deficiencies of both B cells and T cells, or only of T cells. In the later case, the defect in humoral immunity is due to absence of T cell help. Children with SCID usually have infections during the first year of life. Then DiGeorge syndrome, it is congenital. Thymus gland is absent or underdeveloped at birth. Problem with the T cell maturation. Usually it is due to chromosomal abnormality, but it is not inherited. Reason for disorder is not known. Boys and girls are equally affected. Then Actinic DiGeorge syndrome, sporadic microdeletion of 22q TBX1 gene, TBOX1. Then lack of T helper cells, cytotoxic T cells, CTL, and T regulatory T reg cells. These cells are present, but T-dependent B cell response are defective. Then antiviral and antifungal immunity impaired, developmental defect in the third and fourth pharyngeal pouch. Results in facial defect and congenital heart disease. Then Chidiac-Higashi syndrome, I'm not sure about the pronunciation, or CHS, we will be shortly representing as CHS, is a rare inherited complex immune disorder that usually occurs in childhood, characterized by reduced pigment in the skin and eyes, oculocutaneous albinism, immune deficiency with an increased susceptibility to infections, and a tendency to bruise and bleed easily. Mutations in the CHS1 gene, also called LYST, located on the chromosome 1q42 to q43, have been found to be connected with this disease. This gene providing instructions for making a protein known as the lysosomal trafficking regulator. Researchers believe that this protein plays a role in the transport of materials into lysosomes. Lysosomes act as recycling centers within cells. They use digestive enzymes to break down toxic substances, digest bacteria that invade the cell, and recycle worn out cell components. Although the lysosomal tracking regulation protein is involved in the normal function of lysosomes. The next, vaccines. Vaccine is a biological preparation that provides active acquired immunity to a particular infectious disease. A vaccine typically contains an agent that resembles a disease-causing microorganism and is often made from weakened or killed forms of microbes. Its toxins or one of its surface proteins. The agent stimulates the body's immune system to recognize the agent as a threat, destroy it, and to further recognize and destroy any of the microorganisms associated with that agent and it may encounter in the future. Then types of vaccines: attenuated live vaccines, inactivated killed vaccines, toxoids, DNA vaccines, recombinant vaccines. Then live attenuated vaccines. Virulent pathogenic organisms are treated to become attenuated and avirulent but antigenic. They have lost their capacity to induce full-blown disease but retain their immunogenicity. Then live attenuated vaccines should not be administered to persons with suppressed immune response due to leukemia and lymphoma, other malignancies, receiving corticosteroids and anti-metabolic agents, radiation, and pregnancy. Then inactivated killed vaccines. Organisms are killed or inactivated by heat or chemicals but remain antigenic. They are usually safe but less effective than live attenuated vaccines. The only absolute contradiction to their administration is a severe local or general reaction to previous dose. Then toxoids. They are prepared by detoxifying the exotoxins of some bacteria, rendering them antigenic but not pathogenic. Adjuvant alum precipitation is used to increase the potency of vaccine. The antibody produced in the body as a consequence of toxoid administration utilize the toxic moiety produced during infection rather than act upon the organism itself. In general, toxoids are highly efficacious and safe immunizing agents. Then DNA vaccine. A DNA vaccine is a type of vaccine that transfers a specific antigen coding DNA sequence onto the cells of an immunized species. DNA vaccine work by injecting genetically engineered plasmid containing the DNA sequence encoding the antigen against it, which the immune response is sought. So the cells directly produce the antigen, thus causing a protective immunological response. Then surface antigen vaccines. It is prepared by cloning HBsAg gene in yeast cells where it is expressed. So HBsAg produced is then used for vaccine preparations. Their efficiency and safety also appear to be high. Live vaccines, live attenuated vaccines, killed inactivated vaccines, toxoid, cellular fraction vaccines, recombinant vaccines, etc. So if you try to learn that. Live vaccines, for example, smallpox, variola vaccine. Live attenuated vaccine, BCG, typhoid oral, plague, oral polio, yellow fever, measles, mumps, rubella, influenza, typhus. Okay. Then killed inactivated vaccines, typhoid, cholera, pertussis, plague, rabies, salk, polio. Then intramuscular influenza, Japanese encephalitis. Then toxoids, diphtheria and tetanus. Cellular fraction vaccines, meningococcal polysaccharide vaccine, pneumococcal polysaccharide vaccine, hepatitis B polypeptide vaccine. Then recombinant vaccines, hepatitis B vaccine.